Hi, Darshil. Thanks so much for joining me on this episode of Concentrating on Chromatography today.
Thank you, David. The pleasure is all mine.
I'm super excited for this conversation because you have such an amazing story. You were originally trained as a dentist in India. So, what sparked your interest in forensic science and analytical chemistry in general?
I think during my internship, I wanted to do something different from all of my colleagues that were either planning to do a master's or were trying to planning to do a master's in public health or something that was a generic pathway or pick up a specialty. And I looked up forensics. Initially, it was forensic odontology that I wanted to pursue. And from there, we were looking at programs across different countries, and I looked at Australia. And from talking to the universities, the program that I wanted to pick up, it was shutting down that year. So then I looked up at generic forensic programs, and then the UTS forensic program came through, and it looked interesting. And yeah, and that's how I enrolled in the forensic science program at University of Technology, Sydney. And yeah, it wasn't this part was not super planned. Like I wanted to do forensic odontology, but yeah, so that's how I kind of picked up and ended up picking forensics as a stream.
I always joke that I believe that there were a number of people around my age group who became really interested in forensic science due to television shows like
Yeah,
CSI and NCIS because I went to a college where there was a forensic science major. And I just remember that being a
interesting.
major point of
Yeah.
inspiration for a lot of those people.
Yeah. Yeah, and that's always a common starting point. Even when I speak to my students or talk to people, that's what kind of intrigues them. I the only crime show I watched was Dexter. Apart from that, yeah, I didn't watch NCIS, but yeah, it was interesting.
Was there a defining moment when you realized that you wanted to pursue research full time?
Yes, so in my second year, I we had to pick either I we could transfer to like honors program, and I by spending my first year at UTS, we were told that research was important to pursue if you wanted to do forensics. Then it was background would help you in progressing in your career. And so in my second year, I approached different supervisor or principal investigators at the time, and then I got a research project, and we'll talk more about that. But through that, I think in my first 6 months is where the interest sparked in my honors program, where I was like, okay, I'm more interested in this, and was more inclined to then pursue. And then one thing led to another, and in like a year or so, I knew that I wanted to do PhD and move forward in this field.
As you know, this podcast is focused on separation science in general.
Yes.
I'm always interested to hear how people first become introduced to chromatography
Yes.
and mass spectrometry. I'd love to hear that from you.
My introduction was because of my background and the type of research that we did in my lab, which was focused on human decomposition process and looking at how we can locate people using chemical signatures. My introduction was, I would say, not like a generic one where you sign up for some of the chemistry courses, you do chromatography, you do mass spectrometry. I would say my introduction was directly through my research project that I was given. And so I was looking at early postmortem period VOC profile, like volatile organic compounds that were released. or the odor that was released during the time of decomposition. And so through that was my introduction. So I went directly to GC-GC, which is not a traditional route for most of the people, and I consider myself very fortunate. But yeah, that's how the gas chromatography and mass spectrometry was like I came across it through my research project.
And I'd love to keep going with that as far as did you find GC-GC because that was just the best analysis
Mhm.
approach for the work you were doing?
Yes, I feel like it, the lab had already established that the comprehensive two-dimensional gas chromatography was the traditional route for the analysis. So human decomposition order is a very complex mixture of different components, and so it was already by that time established that for analyzing the volatile organic profile released from human decomposition, comprehensive two-dimensional gas chromatography just performed way better than a traditional one-dimensional chromatographic techniques.
And I mentioned to you off camera that I've been lucky enough to learn from some of
Yeah,
the other experts doing
yeah.
GC-GC.
Mhm.
But I'd love for you to try to explain GC-GC as an approach for someone who might just be familiar with LCMS, for example.
Okay. So I think like the comprehensive two-dimensional gas chromatography or GC-GC is built for complex samples. So we have two different separation mechanisms involved that are the two different columns with two different stationary phase coating. They can have different dimensions. So we usually leverage two separation mechanisms. Those are connected by an interface. We call it as modulator. And the purpose of the modulator is to collect the analytes that are traveling from the first dimension column and then refocus them on a second dimension column, which has its own separation mechanism or stationary phase coating. And so it kind of gives you like a complementary, or it enhances the separation from the first dimension column. And so we get better separation, better resolution of compounds. And so we are able to separate complex samples when the conditions are optimized.
Now, I know that people in academia and people doing research tend to move around with some
Mhm.
regularity, but you've
Yeah,
been lucky enough to work in Australia, Canada, the United
yeah.
States. How do you feel being exposed to those areas has helped you evolve as a scientist?
So the one thing I kind of tell people that you never plan for these things. It was never a trajectory that I had in my mind. When I look at 10 years ago, I would have never imagined before going to Australia that I would go to any other country than that. Through different opportunities, I was fortunate to kind of given this opportunity to move from one country to another. But yeah, I would say like it kind of allows you to grow a lot as a person, and you also become aware of your strengths, your weaknesses, and then things evolve from one country to another in terms of the research that you get to do. And yeah,
During every interview, there's always a phrase that I'm unfamiliar with, which I usually have to look up how to pronounce. In
mm,
this case, it's a human taphonomic facility. I'd
yeah,
love to hear about what that is and why it's important for
yeah.
forensic research.
Human taphonomic facility are basically they're colloquially called as like body farms. So here we kind of study the human decomposition or in an outdoor environment. So it's a fenced facility where we basically we do research on different, I would say different areas within forensic taphonomy. So you have The forensic taphonomy itself is the process of understanding death, and the process that kind of allows us to determine the cause of death, the nature, what happens to the remains after postmortem period or during the postmortem period. And so we have different disciplines that are involved. So normally at a taphonomic facility, we have multiple disciplines that are working together and trying to understand how the process of decomposition or death happens in that environment. As the environment or the a certain geographical environment is pretty unique, and it has a lot of impact on the decomposition process itself. So the law enforcement agencies work closely with the researchers, and sometimes we have questions from the law enforcement agencies that we are trying to answer, or something that has come up in a casework, we are trying to replicate those scenarios. And so it kind of allows us to gather local data that will not only be relevant to a certain state or a country, but it also allows us to then look at certain geographical environment and then understand how how that process happens.
And if I understood correctly, you actually worked at the only facilities of this type in both Australia and
Yeah,
Canada. So these are
yeah.
clearly very important, very special facilities. I'd love to hear in general terms about the kinds of analysis that are going on. I think you did a great job discussing some of the general reasons
Yeah.
that they exist.
Yeah. So, as I said, there are different disciplines involved. So, when we were in Australia or Canada, we were looking at the chemical signatures that were released during the decomposition process. So, this was the VOC analysis. Then we also looked at the impact of clothing. So, it kind of changes how the way the body decomposes. Then also we looked at burial scenarios. So this is where burying a person in a shallow grave versus a deep grave. Mass burials or mass graves. So this is where there are multiple remains that are present within or buried within a single grave. So and then from anthropology perspective, so looking at burial practices and how that changes process of decomposition, entomology, so how insects find the bodies as they are most commonly the very first species that are able to locate and track the body. And then different, I think, DNA analysis, fingerprints, recovering fingerprints from post-mortem remains. So a lot of different type of research happening. There's also drones involved, so from search and rescue perspective, how do we look for the bodies or the graves? It, at times, these facilities are located in like forest area or like they're a bit more covered. So you try to also see how we can gather the drone data or the surveillance data when there is canopy involved. So I think go on. The other thing is um, from like the isotope analysis. So this is where you can look at different isotopes that can be detected for the purpose of decomposition, looking at fungal, so different types of fungal species. There are different in Canada versus in Australia. So seeing at the late stages of decomposition how they evolve or which species comes. Dogs, so the training of dogs also happen in different scenarios. There's like a concealment scenario, so in a vehicle. So yeah, there are the multiple disciplines involved. And that's why it creates like a really big research environment where you generate real-time data that can be used by law enforcement agencies to then answer pertaining questions that are most relevant to that particular area, as the environment is different from even in United States, like the North Dakota weather is completely different than Florida. And and so that's why you can generate a really unique data that law enforcement agencies can look at for that geographical regions and take an informed decision if that sort of research has happened. And so that's why in my second year, I didn't knew when I enrolled in my project and during my master's. And going back to your initial question, so this is kind of when it kind of sparked the interest. And I thought, okay, this is what I would like to do and continue to research on.
Now, I'm sure that basically everyone you meet and talk to about your work has some
Yeah,
pretty large misconceptions about the
yeah,
concept of body farms and the research being
yes,
done. What are
yes.
those and how do you deal with those?
I feel like it comes from it because it's such a taboo topic, like the death and processes after it. There are one of the biggest misconception is it becomes very smelly, which I mean our nose after seeing so many decomposition process are not the right. They are a bit desensitized, but I would say like it's not like that. Like it's not like that it's gonna be very smelly or something like that. You barely can notice it from a far distance. So that is one of the big misconceptions that people have. Or it's a disruptive in nature. Normally these facilities are away from residential locations, but they are not disruptive. I would say they are more peaceful and calm areas. So yeah, I think these are the two biggest ones that we normally come across. The biggest one is smell. People are concerned about the smell, or people are concerned about body parts. So the bodies are placed within the anti-scavenging cages. So the the security of the remains is the number one priority, and every tissue, bones is accounted for. Every hard tissue is accounted for because the soft tissue kind of breaks off. But yeah, so these are some of the kind of, I would say, misunderstandings. But I guess it comes from a genuine interest, I guess, because not many people would have come across or in their lifetime of such place. I never came across something like this before starting my master's project. So yeah.
And speaking of odor profiles, what variables have the biggest impact on decomposition, chemistry, and odor profiles?
I would say I think the biggest one is like the sample itself, like each human is very unique. Although we are made up of the same macromolecules, the ratios in which these macromolecules are present within our bodies are very unique to an individual. Our diet, our habit, our physiological process, it is very unique to us. And I think that is one of the biggest variable is the the sample itself. It's not it's no two bodies are the same. They would never be the same. So that's the other one of the biggest the variable that you have to account for. And so there are internal and external variables. I think the external one is, as I said, like the environment is the biggest one. Is I would say like, and this was, I think, my biggest learning from Canada and Australia, like no two summers are same. No two winters are the same, or even like the rainfall we get within summers is so variable. And so the season and the temperature and the environment plays a very big role. But just comparing the climatic patterns and the donor itself, I think are the biggest two variables within. And then certainly there is clothing and other external factors, but I would say like these two are the biggest ones.
what beyond variability makes human decomposition so difficult to analyze chemically?
I would say that because these processes are still from like the biochemical standpoint, because they are not understood, like the breakdown of the cells or the tissue, like they are, we cannot predict at a certain rate. It's where, there are, I kind of explain this to my students this way, that, when you're thinking of a reaction, you control the conditions, right? You have a starting material, you control the conditions, and you get a perfect reaction. But with human decomposition, there is no one way that it will go because the diversity of the human body itself, the breakdown process can happen multiple different ways. Even the parts of the body does not decompose at the same rate. So this is where the face will decompose faster versus the limbs. And so that a lot of variabilities happen within the body itself. And so from a biochemical standpoint, it's very hard to predict and kind of becomes challenging when you're even developing a method because you will not have the same two tissues breaking down in the same manner.
And what are the volatile compounds that are released during decomposition, and how could they impact a forensic investigation?
So these compounds are produced by a couple of different ways. So one of the ways is how the body breaks down itself. So due to just the process of breaking down, the volatiles are released. The second way is through the action of microbes that are present already in the body, and they kind of break down the tissue, and through their metabolism is where the volatiles will be produced. And also, like just the reaction of the body to the external environment will also change how the volatiles are produced or released. But the biggest way that the volatiles are applied is kind of from like the search and discovery point of view. So this is where the it allows us to kind of detect the body or locate the human remains from a forensic standpoint. We have scent detection dogs that we use to kind of locate and search the body. Yeah.
And most listeners to the podcast are familiar with traditional GCMS.
Mhm.
I was hoping you could touch on, for decomposition research, why GCGC TOFMS is so valuable to your work.
Because the diversity of the compounds that are produced as a process of decomposition, we have different compounds that belong to different functional group that are produced as a part of decomposition process, and that creates a very complex profile in itself where we have the diversity amongst the chemical families, but also within the compounds, like within those families, we have multiple compounds belonging to that. So we have different sulfur compounds, different nitrogenous compounds, and so forth. So we have multiple chemical families involved, and within those we have different compounds. And so the profile becomes relatively complex. And so that is why the comprehensive, the GC-TOFMS is used, is because to kind of cater to this different functionalities and different compounds within, and it allows the separation of this complex mixture of compounds and then their identification.
And is there a specific compound or maybe a pattern that would be missed if you weren't using GCGC that's now available because you're using this approach?
Yeah, I would say like if the compounds were co-eluting together or they had similar boiling points or polarities in relation to the stationary phase, then they would get missed if you are just using one-dimensional GC. But in the two-dimensional gas chromatograph, you can really cater to different polarities and boiling points. So that's how it kind of gives you an advantage to separate these out. And that's why for the VOC analysis, I guess that's where the advantage is for the decomposition order, where GC-GC really shines in separating this complex profile.
And when I hear the term comprehensive, I believe that's
Mhm.
connected to the large amount of data that's produced. How do you handle dealing with and sorting that massive amount of data to find the answers you're looking for?
So I think that now the data processing pipelines are a bit more established compared to a decade ago. So we have there are more softwares available too, and also some of the open source platforms now that you can process your data in R or Python or using a Python script. But I think traditionally the best way is to use a software or a vendor specific software, or the standardized way is to kind of use a vendor specific software that allows you to not only align the data but then also filter the data out. It requires us to kind of collect control samples, your quality control samples, to kind of really parse out the the unique compounds that are unique to your sample or your experimental samples. So it becomes challenging. You see a lot with more power, you have more troubles. But yeah, certainly is a complex process. The vendor specific softwares to help.
And to switch gears into the applicability of your
Mhm.
work, how does the research improve cadaver dog training and human remains
Mhm.
detection efforts?
Yeah, so I think it goes back to, I would say, like the comprehensive or the rich data that is generated with the GC instrumentation. And this is where we can really improve the training of the dogs. We can improve the development of sensors because we will get like a complete picture that might be missed with just using a 1D GC. So this is where I think the applicability lies, is where if the profile or a training aid was developed using a 1D GC compounds, then the 2D um, chemical profile will be much more comprehensive in giving you a complete picture or a complete scent profile of the training aid. And that eventually kind of improves the training of the dogs too.
And I think you just touched on this when you mentioned the sensor, but could you
Mhm.
touch on the concept of the portable electronic nose? And
Yeah,
do you see that eventually maybe replacing the need for cadaver dogs?
I would say like right now, the combination of like a very well trained cadaver dog plus the handler is very efficient and there is no match for it. But I can envision that as we go on this path, and there is certainly a need because there are certain areas where you cannot really send a dog or a hen. There's challenging terrains, areas where you just cannot send a person or a dog there. It's not safe for them, like in search and rescue or a collapsed building. Sometimes it's just not safe. And so there is a need, yes. And I think it will, as the sensors develop and as like our understanding of the chemical profile involved evolves, is where we will get to a stage where we will have sensors that will be used widely. But at this moment, if you would have to ask me, then yes, a really well-trained cadaver dog plus a handler combination is too good. Yeah.
What would you say are the biggest challenges facing forensic VOC analysis and decomposition research?
I think the biggest one is just the sample itself. I would say as we move on from like in last decade, as let's say that we have laid a foundation, we have now understanding of the VOC data from decomposition, or decomposing remains. I would say the next stage is to just have bigger studies with multiple donors, and that is challenging also because I would say that that is the biggest bottleneck, or that is the biggest challenge, is to have multiple samples to get around this uh, inherent variation that we have in the sample itself, right? And that is kind of the biggest one. The second one is, I think, accounting for environmental factors. It kind of is a double-edged sword, right? Like we say that it impacts a lot of the decomposition process. And so we need more data from different geographical environments to kind of then really have that understanding. If we envision that this chemical profile is then widely applied across the world or in different environments, then that understanding needs to be standardized, or at least have a complete picture for different geographical locations. Yeah.
And I understand that you're building your own lab at the University of
Yes.
North Dakota. I'd love to hear
Hmm.
about what you're most
Yeah.
excited about as far as the direction of your research and then even where GC is headed in this area over the next decade.
Yeah. So my lab, I'm really excited that I would have uh, my lab would have like on GC. And I think the couple of detections is one, trying to introduce GC to some of the traditional analysis that have been performed on 1D dimensional, on 1D GC. Also look at um, the use of hydrogen and its applicability in forensic labs. So I think these are the two big areas where, and also look at some of the complex samples. So doing research that is relevant to North Dakota. I think this is where, again, using like GC in the areas that have not been really, the GC is not applied to the samples relevant to North Dakota. So really excited to kind of start this journey.
That's great to hear. Is there anything else about your work that you'd like people to know who are just learning about this area of research for the first time?
Yeah, I would say that, it's still like an evolving field and there's a lot of work to be done. So say like more replication across different labs, or just the inter lab studies that are very important, so that there could be some form of standardization that comes through. So inter I think inter lab collaboration research for the same area, I think, would also add value. And we have these missing pieces of information, so it will kind of help us to make a complete picture.
Well, Darshil, I really appreciate your time today. Your story is super interesting, and so is this research.
Thank you, David. It was really a pleasure to speak with you. I think I've never been able to share some of the information that I did today, so it was good to kind of talk to you and share this with the rest of the world. Yeah.
Happy to hear that.